Hello students. In our today's lecture we are going to learn about the one of the plant pathology topic that is called the mycoplasmal diseases. So in this our today's lecture the mycoplasma what here disease that organism cause the disease on the plant, so that we are going to learn, right? So in this lecture we will start about the different kinds of plant diseases. That is one of the mycoplasmal disease, that is called the grassy shoot disease of sugarcane, right? So this disease caused by the mycoplasma on the plant, that is called the sugarcane plant, right? So what are the significance of this disease, means why we have to study this disease, right? It causes the several damage to the sugarcane crop. So this grassy shoot disease is one of the most important disease caused by the phytoplasma, right? It is one of the mycoplasma. So it is observed in many states of India, like the Maharashtra, as well as the Madhya Pradesh, Uttar Pradesh, Karnataka, Gujarat, right? So in this region where the sugarcane cultivation, as well as Tamil Nadu also, Andhra Pradesh also, so here this disease get found in our Indian states. So this disease, it causes the severe damage to the raton crops, that is the sugarcane crops, right? So we will see the economic losses, what economic losses caused by this disease. So it reduces the germination of seeds from the disease canes. So if it will not going to that is the disease cane not going to germinate, then what will happen? Plant will not going to develop, and if the plant will not going to develop, there will be severe economic loss for the farmers. So it shows the stunted growth and poor juice quality of sugars. If there will be a poor sugar quality, juice quality, then what will happen? Ultimately the crop production will going to lose, as well as the rate whatever it will that plant will going to, that is sugarcane will going to get the rate, so that will be again get reduced because of this disease. The infections up to 10% do not cause significant reduction in yield and the number of malleable canes. But if the disease will get severe, means it will, the infection will be get spread out rapidly and it will be more than 10%, then there will be 100% reduction in the yield. So the threshold level for the yield reduction is near about 15% infection. So if the 15% infection or more than that will be get, then there will be threshold level of yield reduction of sugar canes. The distribution. So this disease it was first noticed in India in 1919 in the Maharashtra, right? So subsequently it found it was in Andhra Pradesh, Tamil Nadu, Orissa, Bihar, Uttar Pradesh, Punjab, and Rajasthan also even. If I am talking about this disease out of the India, right, outside the India, then it's reported from the Myanmar, Sri Lanka, and Sudan, right? It means it's this disease is spread out even in the out of India also. What are the symptoms of this disease? The first and the major symptom of this disease is the profuse tillering and the greasy appearance of the stools. The shoots growing from the disease sets, they remain stunted. Stunted means their height will not going to be normal. It will be get stunted, short height they will get. So the leaves are narrow and small like the grass leaves, right? So sugarcane leaves, they are very larger in size. They are longer as well as they are wider also. But if this disease will get, then the leaves of the sugarcane will be like grass, means they are smaller in size. The sugar canes, they are thin with the short internodes, right? So if the internodal region will be get increased, then what will happen? The height of the plant will also going to increase. But if it will be infected by this nematode mycoplasmal disease, then the internodes, the internode means the region in between two different nodes. So it will be going to shorter, and thus plant height will get reduced, and ultimately the crop it will also going to happen. So giving, so this, that is the short internodes, it will results in the bunchy or grassy appearance to the clumps, right? So these are the major symptoms shown by the infected plant. The leaves of the infected plants, they does not produce the chlorophyll, and thus they will become the yellow, and therefore appear like the white or creamy yellow color, right? So generally the leaves get green color because of the chlorophyll pigments. But if there will be no development of chlorophyll, there will be no production of chlorophyll in the leaves, then what will happen? Then the color of the leaves will going to change, like the white or creamy yellow. The leaves, they appears yellowish, and in some cases may be entirely devoid of any pigments, means they will be completely white, there will be no pigmentation in such kind of leaves. So these symptoms can be seen prominently in the stubble crops. The lateral buds sprout before the normal time on the growing grains. So the lateral bud formation, so they will be early sprout, right, as compared to the normal growing time of the sugarcane. So here you can see the grassy shoot disease in the sugarcane. It's clearly visible how you can identify by the observation of the color of the leaves. So here you can see there is a complete white or pale yellow color of the leaves, right? So in this way you can identify it. Here also, here you will get, there will be a complete white color of this sugarcane leaves, right? So in this way you can identify the symptoms of the grassy shoot disease. Then we'll talk about the systematic position of this mycoplasma, right? So its domain is the bacteria, means it's one of the bacteria. The phylum, it's Tenericutes, and the class is Mollicutes. Order, it's Echoleplasmatales, and the family Echoleplasmataceae, and the genus Phytoplasma, and the species is Oryzae, right? So Phytoplasma oryzae is its scientific name. The causal organism, it's caused by the phytoplasma. It's one of the destructive pathogen of sugarcane, right? As it completely reduces the crop yield of sugarcane. The phytoplasmas are spreading at an alarming rates and adversely affecting the yield of sugarcanes, right? So this phytoplasma, it formerly called as a mycoplasma like organism, but after that scientists have shown it's under the electron microscope and find its structure, and then they declared that it's a mycoplasma, not mycoplasma like organism, right? So it's large group of obligate intracellular cell wall-less parasite. It means generally bacteria having the cell wall, right? But this phytoplasma, it doesn't have cell wall. It's a parasitic in nature. It found in the inside the cell. It means it's a intracellular parasite. Sorry, outside the cell. That is intracellular parasites, and it is obligatory parasite. It's found in the sieve cells of infected plant. So as they are found in the sieve cells, sieve cells generally their food material is there, and if this phytoplasma it will be infected, then all the food material it will going to obtain from the host tissue. So there are two types of bodies which are noticed, that is spherical bodies of near about 300 to 400 nanometer diameter, and they are filamentous of 30 to 53 millimeter in diameter. So these are the two different bodies of this pathogen which are noticed. How transmission will be takes place? Transmission means from where the plant will get infection of this mycoplasma. The grassy shoot disease is primarily transmitted through the diseased seed material. If the seed will be get infected by this mycoplasma, then 100% the plant will also going to infect, and perpetuated through the ratonin, right? So this disease is also transmitted by mechanically, by cutting knife. If the cutting knife having such kind of bacteria on that is mycoplasma, then plant will also going to infect by mechanically. Or some insects, that is the leafhopper, okay, or aphids, so they act as a vector, right? You know, the leafhoppers as well as aphids, they feed on the plant leaves, right? And ultimately as they are vectors, so they will transmit this disease, transmit this mycoplasma to the plants, as like we get the malarial infection from the mosquitoes, right? As well as the dengue also. So as like that, the leafhopper as well as aphids, they act as a vector to transmit such kind of disease, as well as the dodder also, okay. The aphids, that is the Melanophis sacchari, it's generally transmit this disease. The optimum acquisition feed period is near about 15 minutes, right? If aphid will be feed on the plant up to the 15 minutes, then plant will going to infect by this mycoplasma. The single infected aphid can transmit the pathogen, right? Optimum number is 15. The transmission is non-persistent manner, right? So there will be no any kind of persistent, it will be a non-persistent manner it will going to transmit. The pathogen has been transmitted from the sugarcane to the sorghum, and from sorghum to the sugarcane by using the aphids, right? So there will be a cycle of this disease, right? So if the aphid will be feed on the sugarcane, so then that aphids will be transmit such kind of disease from the sugarcane to the sorghum, and again from sorghum to the sugarcane. So in this way the cycle will be takes place in between these two different plants. So sorghum is also the natural host of GSD phytoplasma. So this phytoplasma is also cause the disease on the sorghum also. Then how we can control this disease, how we can manage this disease right now? So that is called as integral disease management, IDM, okay. So the phytosanitary controls measures we have are very important to control such kind of diseases. If you are using the knife as well as the cutting scissor, right, then you have to sanitize such kind of instrument before the utilization. Otherwise, if it will be get infected by such mycoplasma, then you are cutting the leaves as well as cutting the stem of the sugarcane, then 100% the plant will going to infected by this mycoplasma. There are some growth resistance varieties of sugarcane, like the CO 86 to 49 or COG 93076 or COC 22. So these are the three different mycoplasma disease resistant varieties. If you are using these varieties, then mycoplasma cannot be infect such kind of varieties of sugarcanes. The use of healthy diseased free planting material collected from the seed nursery for the planting. Okay, so when you are using the seeds for the plantation, so you should select such kind of seeds as well as the planting material. It should be healthy, right? When you are multiplying it under the nursery, right? The heat therapy, it means you have to heat the seeds or the planting material should be treated with the hot water, and that hot water temperature should be 50 degrees Celsius for the 120 minutes. Means you have to keep uh, such kind of seed or the planting material in the water for the 2 hours, so that water will having the temperature of 50 degrees Celsius, or the moist hot air of 54 degrees Celsius for the 2.5 hour have to be get uh, such kind to avoid such kind of uh, infection, right? So that will eliminate the pathogen from the diseased seed material. Okay, so if you are suitable with the hot water treatment, then just go for it. Or if you have some advanced instruments like the moist hot air, then you may go with this because it's a more prominent way, right? So this is heat therapy is also one of the good choice for the control of such disease management, as well as the rattening of the affected crops, right? So it should be avoided, otherwise disease will be spread. Crop rotation may be employed to reduce the inoculum in the field. For example, in this year you are cultivating the sugarcane, then next year just go with the groundnut or any other crops. So this crop rotation is beneficial, right? Because this mycoplasma cannot attack on different plants like the groundnut. Then just go with if in the next year just go with the groundnut, in the next year just go with the wheat, and in the next year you can cultivate sugarcane. If there will be a gap, then such mycoplasma which is present in the soil as well as in the field, right? So when it doesn't get housed, it will going to die, right? So in this way you may have to rotate the crop. So it's very good disease management to control such kind of infection of mycoplasma. The rogging of affected stools and their destruction, that is the mass eradication, may help the consistent reduction in inoculum in the areas. So if in your field, if some of the plant will get infection, then just uproot that plant and burn it, right? Then this will be also beneficial. The control of insect pest, you know whose are the vectors for this mycoplasma, the aphids, right? Nematodes, uh, grasshopper. So such insect you have to control by applying the insecticides, right? So that this mycoplasmal infection to the sugarcane it will not get, right? So this is also one of the kind of good method. Then set the treatment with the 500 parts per million solution of lidermycin. It's an antibiotic before the planting. So before planting, what you have to do? You have to treat either seed or the crop part which are you are going to utilize for cultivation. So treat with the such kind of antibiotic, that is the lidermycin, with the 500 ppm solution. So that will be beneficial, as lidermycin will going to inhibit the growth of such bacteria. It will, so bacteria with such kind of bacteria, not bacteria, it's actually mycoplasma. So this mycoplasma growth will going to reduce, or it may get mycoplasma may get dead because of this antibiotic. So today we'll stop here. Thank you.